The sixth mass extinction is upon us, and it is known as the Anthropocene. A mass extinction is a rapid decrease in biodiversity usually caused by an abiotic environmental factor such as volcanoes or meteors. The Anthropocene is a special extinction because it is currently happening due to a prevailing biotic factor, humans. Humans are causing the sixth mass extinction by implementing habitat loss when clearing land for agriculture. A perfect example of this deforestation takes place in West Africa, where approximately 80% of the world's cacao is grown for chocolate. Of the 80% of cacao produced by West Africa, 40% is grown in Côte d'Ivoire alone. This is where my research comes into play. I am Rachel Kreitz, and this summer, I am working in Dr. Noé de la Sancha's lab, taking part in an REU internship program at the Field Museum of Natural History in Chicago, Illinois, to study a species of small, cute, furry mice called Praomys. Praomys calls Côte d'Ivoire home, and sadly its habitat is being destroyed to support the world's high demand of chocolate. So how will my research support the conservation of these small yet ecologically important mammals? There are two questions I must answer to help minimize both habitat and overall biodiversity loss. First, what type of variation exists in Praomys rostratus? Is variation present in sex, age, or habitat? Second, how does the ontogenic variation of Praomys, or the way the mouse morphologically changes with age, compare to other rodents around the world? Ontogenetic variation can be defined in three parts: growth, development, and allometry. Growth is a change in size as an organism ages. Development is a change in shape as an organism ages, and allometry is a change in shape as size changes. This research is significant for two reasons. We will be able to more accurately describe the number of species in the tropics, which for small mammals is practically non-existent, and we will be able to document how small mammal species from the tropics change over time. To answer these two questions, I implemented geometric morphometrics to measure the skull and mandible shape of Praomys rostratus. Traditional morphometrics use calipers to measure the shape of a skull the same way a ruler would allow someone else to measure the length of a straight object. Geometric morphometrics used for this project were obtained through computer software that quantified the skulls and mandibles. Using geometric morphometrics helps to better visualize these shapes through models. I then use these models to quantify shape and run statistical analysis comparing variation in sex, age, and habitat. The 5 microhabitats tested for variation include cocoa, fallow, rubber, primary forest, and secondary forest. My results suggest that Praomys rostratus does not display sexual dimorphism, but does exhibit ontogenetic variation within microhabitats in Côte d'Ivoire. Biodiversity, by definition, is the variety of life on Earth. Obtaining ontogenetic variation will allow me to quantify the most basic unit of biodiversity, which is species and their variation. Biodiversity loss is inherently linked to deforestation. Eliminate the habitat, wipe out the species, lose the biodiversity. It's as simple as that. It is important to study the development of organisms like Praomys to understand how change in ecology, such as massive habitat loss, can affect conservation and overall biodiversity loss. Habitat loss may be reversible, but biodiversity loss is forever. That is why it is more important now than ever before to learn, understand, and apply coexistence strategies with native wildlife in biodiversity hotspots like West Africa. The Anthropocene is not permanent, because habitats can still be protected or restored. By collecting data on Praomys this summer, I help add to the information that supports conservation strategies developed to save biodiversity in West Africa. Chocolate may be pretty sweet, but saving species like Praomys and the unique habitat of West Africa is even sweeter.